Okay, welcome everybody. It's Friday. Let's start because it's time. I see still two people missing, but I'll have a look in a moment who those two persons are. So welcome. Today it will be all about finishing up these genetic things and making sure that we understand everything well, because next week we will have a close look into inheritance, and to understand inheritance, of course, the genetic concepts are of utmost importance. So what are we going to do today is first I will go over the content of Canvas, which is basically things that you have already seen. Then I will have a quick look at how genetics is done today, because in fact it might come to a surprise to you, but genetics nowadays is not that much putting liquids together in the lab anymore. Or it's also that, but it's a lot of bioinformatics as well. So genetics is because there is so much data. If you want to become a geneticist, you really need to deal with informatics, be good with certain computer programs, and I want to show you a little bit what those scientists use to do that kind of investigation. And then finally, we will do the activity in Canvas we have together. Usually I can finish it. In the other group, I almost finished it. Yeah, that's it, in fact. But before we start, we will do the quiz, what I told you about on Canvas. So the one you have to do today is the following one. I don't think I have it open. Let me look for it. It's like genetic concepts, the name, but I think I closed it already. I'm gonna have a look for it for you to show. No, I closed it, yeah. I'll open it again. And you have to do that one. I'm gonna give you 2 attempts, but then the second attempt of this quiz is gonna be next week, because in this quiz there is certain concepts that are really part of inheritance already, because it mixes the two things. But I would like to do it right now already for you to understand for yourself how much you understand, and a quiz sometimes helps to make some concepts a bit more clear. So that's why I think it's a good idea to do it twice, this quiz, starting today, to see how far we are in our understanding of genetics. So we are here in genetics week 11, and what I would like you to do is this genetic concepts review questionnaire. So I'm going to give you some time to do that, and then once you're ready, we will come back to this. Okay? So go ahead. I'll have a look when you finish. So you get like 10-15 minutes to do. Be okay. And if you do bad, it's not a problem.
What is it called again?
The genetic concepts review questionnaire.
Mine says I already have an attempt. It says I have an attempt of like 20 minutes, and then it says 0 out of 100.
No.
I don't know if I can do it again. I don't know. This happens sometimes.
You can. I'll give you a second attempt. 1 second.
Okay. Thank you.
Okay, you should be fine to do it now.
It is the genomics review questionnaire, or which was it?
I think you have your mic turned off.
Yeah, sorry, I tend to do that. It's called Genetic Concepts Review Questionnaire. I mean, I'll show it. There you have it.
Okay, thank you.
Teacher, can I change again from device? The thing is that I'm on my phone.
Yeah, of course. Go ahead.
I just finished exam. Like,
Yeah, go ahead. I don't know if I have. Yeah, I know it's not silenced. Yes, sure. Go ahead.
okay, thank you.
Teacher, the second attempt will be next week, right?
Yeah. Then we have seen more things, and then it's possibly better to do it with a different knowledge behind.
Oh yeah, so for example, like the laws of segregation, we're gonna see that, right?
Yeah, but we have already seen it. We just know it under a different name. In fact, the loss of
Okay.
segregation is in fact the idea that in meiosis, sorry, that each sperm cell and each egg cell has different a different DNA, and this is the second term of segregation as well. That this diversity is coming from genes switching independently from one chromosome to another chromosome. So it has to do with that. It means that it says something about the first segregation and the second level segregation, right?
Yeah, that's what I got confused with, like not knowing which one was like the first and the second.
Yeah, you already know, but you know it under a different name, in fact. So that's why I give you the next chance next week. Okay, so the first, I mean, I cannot already explain it. The first segregation means basically that each sperm and egg cell basically inherits half of the DNA of the normal one, okay? And it segregates independently. And the second level of segregation just means, has to do with crossing over, you know, that one gene can switch to another, to one sperm cell or one egg cell independently of another gene. So it refers to the fact that it's absolutely random. You cannot say, the whole chromosome will mix, or it's always this gene that crosses over, or always that gene. No, it's completely separate. That's it, referring to that. So in fact, you know already. And you can deduce it from the answers as well. Okay. So everybody's finished, so I would like to continue. So the first thing I would like to do is go to the content of this genetics, just to overview what's in there, because in fact that's why I shared the presentation with you of genetics, because it's really little information there is in here. So if you go to week 11, content genetics, you get a quick look of these cases. So you have here about chromosomes. We have seen that. There's just two new terms that have to be introduced, and it's called haploid and diploid. So what does that mean? Diploid means there is two of them. Haploid means there is one of them. So basically, all the cells of your body are diploid because you have the double amount of chromosomes. The only cells that are haploid are which ones?
Gametes.
The gametes.
The gametes, the sperm and the egg cells, correct. So that's these two concepts. Diploid, all the cells in your body are diploid, and one chromosome is coming from your mother and the other one is coming from your father. We say that they're homologous chromosomes, okay? So you see, you have two green ones, two blue ones, and two purple ones. But you also see that those homologous chromosomes, they are slightly different colors. That's because they're not exactly the same. And that's the thing. If there's very few people with their camera on, but who has like little clear eyes, blue eyes or green eyes or something like that?
Itzel.
Itzel. A ver tus ojitos, Itzel, ¿dónde estás? Ah, she's not here, is she? Anyway, Itzel might have a little bit color in her eyes, but that doesn't mean that her father and her mother have colored eyes, okay? For me, I have blue eyes, but in fact, my mother and my father have blue eyes, so that doesn't really count. But the thing being is that you have a double amount of genetic material, but only one gene of each is expressed, okay? So if Itzel has green eyes, it doesn't mean that her father and mother have green eyes. It means that maybe her mother has green eyes and her father has brown eyes. And and the fact that she has green eyes means that she has the gene of
But
mother expressed. Yes, Annabelle?
aren't they passed on from like the third generation?
Not always, no, no, not necessarily. They can be passed on to third or the fourth generation, but basically we all have genes in our body that are not active, okay? The fact that you have blue eyes or green eyes is just an example, but it means that you have the gene of your mother activated if you have the green eyes and your mother has green eyes, and if your father has brown eyes, then that gene is dormant. It's not activated. And that's why that refers to this concept. But we will see that next week in more detail. Don't worry about it too much yet. So this is what we call homologous chromosomes, okay? They're homologous because they're very similar, but they're not identical. Because like you can see here, the gene might have the same function, but it's not identical. That's, for instance, I had a good friend, Michael, and Michael has a sister, and yeah, his sister's name is Laura, and Laura passed away several years ago because she had cancer. So what happened to Laura is Michael is perfectly healthy, but Laura, she got a bad gene from her mother, a cancer gene. Their mother also passed away, so they're both deceased. And Laura had the same problem, but Michael doesn't have the problem because in his body the gene of his father is activated, so he doesn't develop the cancer. So they both mother and daughter had brain cancer because they got active the gene that was passed on from the mother to Laura. Okay, so that's basically what you see here. So two chromosomes, the blue one from their father, let's say, the purple one from the mother. And if you get the bad green gene, you can have the cancer. And if you have the good gene, then it's okay. But now science is trying, if you have a bad gene, is there a way we can silence the bad gene and activate the good gene? Maybe there is a possibility of doing that. We are looking into that. And why do we have two genes if we only use one? It's because then you have a spare copy. So there is very important mechanisms in the body that if one gene is not working, the other gene is automatically activated. And this is what we try to harvest right now to cure certain diseases, you know? We have double copy, not only for genetic variety, but also to have a spare copy in case the other one is not functioning, okay? It's very important, this. In cancer especially. And sister chromatids, yeah, this is basically a single chromosome. When it splits, you get like a double chromosome. This happens in the S phase. However, take into account in the S phase, chromosomes do not exist. It's just the DNA that is duplicating, okay? Okay. The other thing I wanted to show you is briefly, I'm going to show you how it looks like. There is websites online, and this NCBI is one of the most famous ones, and that I also used to use a lot during my research. So there's a website, and I'm not gonna, so you look the gene here and you can look for it, but I'm not gonna do it again because it takes a long time to charge because there's so much information behind it. So here the TP53 gene is a gene that is very often involved in cancer. And you can look for that. For instance, you can also look for Homo sapiens, mitochondrial gene, or ACE2 gene, or HBB gene. You can look for everything. You can also find E. coli genes, which is the bacteria. I mean, they will write it as Escherichia coli, you see. This is bacteria and virus genes. So it's a website where you can find all kinds of genes, and the information of the gene is nicely written out. So here you have the gene symbol, the P53. We call that TP53 or P53 gene. And what is really now the genomic context. So here, this is what I told about. Where is the gene located? So the locus is 17p13.1. So this means the gene is located on chromosome 17. The chromosome has two arms. The upper arm we call the P arm. The lower arm we call the Q arm. So this is the P arm. It's on the top, and it's in position 13.1. So it's like the coordinates, how how far it's away from the centromere. The higher the number, the further away from the centromere. Okay, so that's how it works. And then here you can see to the gene itself. What you see here is all kind of different types. Okay, it's the same gene, but the gene has different variations. You know, just like you can have a gene for eye color, and you have like a whole different kind of variations for a certain gene. And you can click on it, and you can see where the variation is. You don't have to understand this, guys. It's just to show you how nice we have this bioinformatics nowadays. If you're really interested in it, you can look for everything. So this is the messenger RNA has this code. It's messenger RNA tumor protein. You have this variant. It's like, how long is it? NT. This is 19,149 NT. What would that mean? You know that? Is the length, how many nucleotides it has. Okay. If you go to FASTA, you can download the sequence. So if you go FASTA, you click on it, you download it, and you have to, I already downloaded one, but you have to, you need to, I don't have the program installed to make it visual. But then basically, if you download it, you download the program, you can have the whole sequence. And it's like screens and screens and screens and screens and screens. It's very long, full of letters. All the genetic code is there. So, I mean, you don't have to understand this, but there is so much information in this, and human and scientists, they use this really for a lot of things. So you can do a lot of things with it. And if you like this kind of tools, and if you like data analysis and comparison, this is something you should definitely go into genetics. I've always been very fascinated with it. And here you can see this gene, for instance. This is also important because not all the genes are expressed in all the cells. Okay, there are certain cells that have expression of a certain genes, and other one that is dormant. So if we take this tumor gene, which we call P53, you can even have a look how often you have expression of this gene. And this is very interesting because if you look at that, you can have a look at where the cancer will develop. So if you have a cancer that has the P53 gene involved, this cancer will most likely most rapidly develop in areas where you have more expression. And this is in agreement with what we see in reality, because in the lymph nodes, you very often have cancer development. You have also in the matriz or the endometrium, this is the uterus basically, you also have more cancer development. Okay, for this specific gene. You know, if I take like other genes, it will be different. So this gene, you will not see it very often in pancreas cancer or in other things, but you see that it might be in the bladder, in the spleen, even in the skin, but the highest is definitely here, the lymph nodes. And this is interesting because this is really a gene that is very often damaged in different kinds of cancers, in fact. So, in fact, you can do science yourself if you want. You just have to learn a little bit how to read and to understand it. But it's really nice if you go into bioinformatics. So by understanding what you look at, this is a whole other course, and it's called bioinformatics. And you learn how to use this kind of program and get information out of it. You can go to the gene banks and find similar genes in rats and mice and everything you want. So it's really cool. I don't know if you like this, but I think it's fascinating how we do that. So I just wanted to show you, this is nice. This here you can see, in fact, the genetic sequence of this gene, okay? It's not that long, in fact. I thought it would be longer. So here you see the amount of nucleotides. It's 19,141 nucleotides long. And basically you can copy all of this, and this is the genetic code of that gene. And basically what you do, if you, scientists, they use these databases because, for instance, you go to an oncologist and they take the sequence of the gene that is affected to you, you're going to compare it with the information in the computer, and you're going to see, okay, the problem is here, this G should be a C or whatever, and then you can find certain mutations and stuff. So this is really cool. And if you bioinformatics is definitely something for you. So to finish the class of today, I'm going to close this. It's too complex to understand for you right now, but if you want to start playing with it, little by little, you will notice, you will understand more. So now this one I want to do together, because I think it's important that I am aware of how much you understand of these genetic concepts that we have seen. Okay, so it starts really easy. The basic unit of inheritance, how do we call that? You, by the way, it's good idea that you participate, because you have to upload it as well. Okay, so we do it together, but it's a homework for you to do in case we don't finish. Yes, dear?
I can't download the file. Like it appeared that it's an error or something like that.
Can somebody else try? If you can open this file.
I already opened it and I had no problems with it.
Yeah, me too.
There's the children, it's children attack in the house. So somebody else can open this file or not?
Yes, Steph and I already opened it, and we
Okay,
had.
perfect. So could you send it to this person that couldn't open it, please? That she also has it. I didn't see who it was, but please share it with this girl that she can also hand it in. Okay. So the basic unit of inheritance is called what?
A gene.
Yeah, gene. Yeah, exactly. The gene, I would also say genotype is okay, because I mean, if it's one unit, it's a gene, but the genotype is basically everything you inherit. So if it's a unit, it's gene, but the basic thing that causes inheritance is all the genes, so that's genotype. So here, the most correct answer is definitely gene, but genotype is also fine. So genes are formed by what?
Nucleic acid.
Nucleic
Nucleic
acids.
acid.
Okay, well done, indeed, nucleic acids. This one. And then we have seen this as well, and I think you have seen it in the video as well. No, in the video it didn't come, but we have seen it before. This Chargaff scientist, what did he discover? He discovered certain proportions of nucleotides to be equivalent, you know? So what would be the correct answer here?
A and C.
A and C are correct.
Yeah, exactly.
Letter E.
He saw that adenine paired with thymine and guanine with cytosine, but he discovered that because he noticed that guanine and cytosine, they always had more or less the same percentages, and adenine and thymine also, so that they happen in the same proportions, and that led him to believe that indeed they paired with each other, because if you have the same amount of of two different kind of things, there might be a connection between them. Okay, how many genes are in the human genome? There is a lot of discussion about this, and scientists they still change it every year, let's say, but more or less
90,000?
No, it's not that much. It's about 25,000. So they have been saying 30,000. They have other scientists in the beginning, they thought it were as little as 3,000, but now there is more consensus with 25, 30,000. Okay. Okay, and then point out the subsection that continues the series nucleotide DNA gene. So what would that be?
Genome.
Genome.
Nucleus.
Because the genome is made out of genes, so that's the next thing that follows. The next thing is the nitrogenous base that constitutes parts of RNA sequence, but not of the DNA. So what is part of RNA?
Uracil.
Uracil. Uracil.
Yep, exactly. And then indicate which of the following molecule breaks the regularity. It's more difficult. With
Proline.
regularity, we mean,
Proline.
Proline.
yeah. Proline is more protein-like. It's amino acid. It's not part of these ones, because all of this is part of DNA, and proline is amino acid part of DNA. So point to the word whose meaning is the closest to or being a synonymous of codon, a synonym of codon.
Letter C.
Yeah, a sequence of 3 nucleotides. Remember the thing that I showed you, that you have the first base, the second base, and the third base. And the combinations of those 3 bases code for a certain amino acid. And by connecting the different amino acids together, you basically get a protein. So it's definitely the sequence of 3 nucleotides. The first one is not correct because it's not a pair, it's just 3 nucleotides. The combination of messenger RNA, ribosomal, and transfer RNA is also not correct because they help in the translation. It has nothing to do with codon, okay? So it's definitely C. Which of the components does not correspond to the series?
Protein.
Yeah,
Protein.
because all the others have to do with the genome. And if you look at this sequence, is this sequence what? Yeah, it's very easy, I think.
DNA.
DNA, and why is it DNA?
It doesn't contain. And I was thinking like it doesn't contain the U.
Yeah,
Yeah.
it has a T instead of a U, so you know it's DNA. It's a piece of DNA. It's not RNA. So this one's more difficult. The feature of the DNA double helix structure is that both strands run in this position. So what is the correct answer?
Circular?
Anti-parallel.
Circular would be for bacterial DNA. That's not the case in human DNA.
Anti-parallel.
Anti-parallel, yes, because the 2 strands are complementary. One runs like that, the other one runs like that. And horizontal, vertical, you can never say that. We will see that also next semester, that we have medical terms to indicate how something looks like. Horizontal and vertical is very relative because it depends on the position of the cell, so that's not valid for us. Perpendicular is also not the case because you know that the helix is combined, it's twisted, so there is not perpendicular, it should be anti-parallel. Nucleotide to nucleic acid is what an amino acid is to what?
Proteins.
Yeah, okay, good that you know that one. And in the messenger RNA, the information is encoded in 3 bases sequences, and each of these triplets is called what?
Codon.
A codon.
Codon.
Just saw that one. The sequence of codons determine the sequence of what in the protein synthesis?
Amino acids.
Yeah, exactly. So it's the codons that define which amino acids it's going to be, and the protein is just a combination of the different kinds of amino acids, and depending on the sequence, how this is built up, this is the kind of protein that you get. So if we talk about transfer RNA, transfers a specific something from the cytoplasm into the something during protein synthesis. So what would that be?
Amino acid slash ribosome.
Yeah, exactly, because the ribosome is involved in this. So it's gonna, the transfer RNA brings the amino acid to the ribosome, and the ribosome is in fact on top of the messenger RNA and is in fact building this protein in or outside the endoplasmatic reticulum. So what is the name of the process of copying a DNA molecule that results in 2 equal DNA molecules?
Transcription.
Transcription. So when is this happening, transcription?
Where or when?
When?
In replication.
During, yeah,
Yeah, but when
during protein synthesis.
when is replication happening?
In the S phase.
Exactly, in the S phase of the cell cycle. Which
Could you also answer their replication?
In fact, I answered wrong because it should be, it is not transcription, it's replication. I did it wrong. Transcription is when you make messenger RNA. It doesn't go away. No, I was confused. It's definitely replication, and replication is happening in the S phase. So it has, replication has nothing to do with transcription. I'm sorry that I mistaked. I didn't even read it, in fact. But it's definitely replication. The replication is happening in the S phase, the synthesis phase of the cell cycle, and the replication is happening because a cell decides to divide into 2, And if a cell wants to divide into two, it needs to duplicate its DNA. And duplication of DNA is replication. Transcription is when the DNA opens and you get a copy of a certain gene into messenger RNA. This messenger RNA travels outside the nucleus and then you make a protein. So basically, transcription is involved into protein building and not in cell duplication, okay? So that's a big difference. Okay, 17. In something, each of the chains of the double helix serves as a template to synthesize the complementary chain. It is common to call this process, I don't know what, because each molecule of something has an old and a new strain. So what would that be?
B. Replication, semi-conservative, and DNA.
Okay, very good. I agree. Because if we read it full, in replication, each of the change of the double helix serves as a template to synthesize a complementary chain. And that's exactly what happens in replication, okay? Okay, 18. What is the process by which a cell converts the gene information in RNA and proteins? So that's the other one.
Translation.
Translation. Yeah. 19. It is defined as a random change in the DNA sequence.
Translocation.
No, a translocation.
Mutation.
It's a mutation. A translocation is basically when this also happens when, for instance, certain people are ill and the chromosomes are not really well formed. You can have translocations, which means that a certain piece of a certain chromosome goes to another chromosome. That are translocations. It's not a very good thing because the cell is really bad. So that's a whole other course, the kinds of DNA damage that you can have. One of the DNA damages can be translocations, other can be mutations. And mutations is very important because mutations is in fact a very serious problem. And we know now that mutations, they cause a lot of problems for everybody. And mutations, you should avoid them. And how do you avoid mutations? Basically, humans get mutations by two main things. It's one, the way how they live, and two, it's also the environmental factors. So if you live in areas with a lot of contamination, those as aggressive contaminants like car smoke or toxic fumes or solar rays, they can cause mutations in the DNA or they can even destroy it. But more importantly, in fact, most cancer is caused by what we eat. I don't know if you know that, but most cancers is really caused by our diet. Not that much smoking anymore because people don't smoke as much as they used to anymore. But anything that is aggressive for your body, and think about red meat, processed food with pesticides, colorants, flavor, chemicals, all these kind of things, they can cause mutation and damage in your DNA. And all what you need in order for having cancer is just one mutation that cannot be repaired by the DNA repair mechanisms. So it's very important that you think about that. So last questions. There are different types of mutations. Which are those that do not alter the body?
Neutral.
B, neutral.
Beneficial or neutral mutations, okay. Beneficial is more for bacteria, in fact, because sometimes the DNA also changes in a good way. This is evolution, in fact. We adapt to the environment we have, okay. So in order to guarantee our survival, in fact, what happens is that our DNA is something dynamic. It should not stay constant. And it has mechanisms to feel and listen to the environment. And the only problem is that that mutations that are neutral or even beneficial, they don't happen in your lifetime. So the benefit is not going to be for you. The benefit is going to be for your children, okay? Because we are unable to change our DNA once we have it. I mean, we can alter it a little bit, but nature cannot do that. Okay, it's in order to generate better offspring. Think about giraffes. You know, giraffes, they came into existence exactly like that because, I mean, they got mutations that created a longer neck, and the ones that had a longer neck, they would survive better. So in fact, it would be a beneficial mutation to have longer necks. So that's in fact how you get a completely different animal, you know, because with the longer neck, they would reach their leaves, and with the shorter necks, they would stay hungry. So that's basically what evolution is about. So a mutation occurred in the following transcription. That is an error in the mRNA formation with a yellow text highlighter mark where the nucleotides changed or occurred, as well as the wrong amino acid in the protein sequence. Okay, so you have to do that. The amino acids, in fact, what you have to do, we're not going to do that right now, but in fact, you need to go to the codon list, because you're not going to know that. I don't even know that. So you have to check AUG, it's coding for this. CAU, it's coding for that. So you have to check. But what we can check here is if you have mutated DNA and the messenger RNA. So where is the mistake? Okay, can somebody tell me?
There were there like 3 T's.
Yeah, there. Like
The
there's
right-
3 T's. Like
The one in the
the
middle
end.
to be a G.
Here?
No, more
The
to the
second to
end.
last one.
Yeah, there's 3 T's.
The one in
And
the
it
middle
should
is
be
supposed to be a
TGT.
G.
So we have 3 T's here, you say?
Yes, and the one in the middle, it's supposed to be a G.
And why is it supposed to be a G?
Because that's the mutated
normal
DNA, and
DNA.
the normal DNA has a G.
Who is saying this? Because I'm not sure with what you're saying.
The one on the top is the mutated DNA.
Yeah.
And the one on the bottom is the normal DNA.
Mm-hmm.
And the one on the bottom has a G instead of a T.
Okay. So if this is the normal one, it's a G, and this is the mutated one, the T is indeed different. Okay. So it means if this is the G, and then this one is mutated in a T, then the messenger RNA is also wrong. Okay, so this is also wrong, and that would mean that this amino acid is also wrong. So you have to see, yeah, already we did it more or less. But anyway, I'm gonna leave this for you to finish, and then you upload it, and yeah, it's almost ready, yeah, right? And that's it for today, and we see each other next week to talk about inheritance. Okay, we're just gonna see the basics like we do always in this course, but anyway, nevertheless, it's interesting, inheritance, because yeah, it's cool. I think it's nice with the tables, and you understand how it works. And of course, to understand all of that, like I said, it's important that you understand these concepts. But I think you did well. I think you really did well, because if you would do it on your own, I think most of you they would know this. And I also noticed that you're a very strong group. I mean, if I see your grades, everybody has really high grades, some a little bit more than others, but yeah, I thank you for being such a nice group, and we see each other next week. Okay.
Thank you,
Thank
teacher.
you.
Thank you.
Thank you, teacher.
Have a good day.
Thank you.